get restarted on uh, immunity in general. All right. Now, you guys probably heard that we have kind of what we call innate or general non-specific immunity and adaptive or specific immunity. So we have two different ones, right? So when we're talking about vaccinations with immune memory, right? You get chickenpox, you don't get it again. That's this type right here, adaptive or specific immunity. Now, all of us have this non-specific immunity or resistance, right? That can include something as simple as an intact skin, intact mucous membrane, or it can actually be cells that are part of it as well. So there's a big range in what we talk about in terms of innate or non-specific resistance, right? Something is very general as having your epidermis being fully intact with no cuts, no punctures, or it can be as almost like the chemical mediators, or it can be cells. So there's a big range of what we consider to be innate or non-specific immunity or resistance. Adaptive or specific immunity, there's not as much of a range, right? That's your T and B cells working together. So then you have long-term memory. So then if you've been exposed to chickenpox as a child, that you'll almost have permanent lifelong immunity for chickenpox, right? We've been vaccinated for polio. You will have immunity from polio almost for the full term of your life. So that's this type of immunity that's specific. The specific immunity is a little bit more complex, much more complex, right? It has the ability to be specific. So the cells that recognize as a chickenpox virus, right? Keep in mind, the virus itself is an antigen. We're not recognizing, and those cells are not recognizing the whole virus. They're recognizing specific proteins on the virus. Those proteins on the virus, as long as it doesn't mutate, as long as the proteins don't change, the cells that recognize those proteins, well, they're kept as memory cells. And then if you're exposed to the same virus without any mutations will have the same proteins. So varicella zoster, chickenpox virus, right? Very little mutations are seen. What happens then? Well, the same virus that we see as kids will usually be the same, almost identical virus. There'll be small changes, but be almost identical to the virus we see as adults. What happens then? Because of very few mutations, the proteins that sticks out of the capsid, found the external shell of the virus, well, the proteins that stick out doesn't change. So then the cells that we saved up as kids because we've been infected with it, well, those same cells become memory cells and they recognize the same proteins because the proteins on the virus didn't change. So then we have that long-term immune memory. Now, viruses that change and mutate a lot, HIV, the influenza virus, the rhinovirus, right? The coronavirus that causes us to have hay fever and colds, right? Not the one that we see right now. Well, those viruses mutate a lot. Because they mutate a lot, there are proteins on the outside shell of a virus, the capsid in the virus. Well, those proteins then start to change over the course of a year. And what happens then? If the proteins are different on the virus, the saved memory cells that recognizes the previous proteins, now those memory cells are kind of useless because now you have different proteins on this new virus coming in. Well, those memory cells recognizes the old proteins, so they don't help and give you long-term memory then. So that's what we're hoping that we don't see with the new novel coronavirus. We're hoping it doesn't mutate. So then the proteins that we're able to recognize, then we're able to form memory cells. Those memory cells can help fight off that coronavirus next year or later on in the fall when we're exposed to it again, because it'll be the same virus, right? Same proteins. So in terms of innate immunity, these are the physical barriers, skin, mucous membrane, even the cilia in the respiratory system Keep in mind, in the cilia and the respiratory system, it has a specific function. Even the mucus in the respiratory system has a specific function. The mucus and the mucous membranes it produces are going to trap any inhaled particles. And any of those inhaled particles, it can, especially if you're outside, you can have exposure to fungi, right, as you breathe in. Well, they should be trapped by the mucus. The cilia then allows us to cough and then move that fungi-trapped mucus out of our respiratory system. So then you can spit it out, you can swallow it down, but that phlegm is no longer in your respiratory system where it can do the most damage. So coughing and sneezing allows us to mobilize the trapped particles that's trapped by the mucus. The particles that are in the mucus can have viruses, it can have fungi, it can have bacterium in it, it can have viruses in it. So as we trap it, the sneezing will move it into our throat, move it into our mouth. The cilia then moves it out, so then we can get rid of it in one way or another. Right? Now, people with cystic fibrosis have a problem allowing them to thin out the mucus. Most of us have been so sick before, and then when you cough, that thick, sometimes yellowish, sometimes green, thick mucus comes out. It's not mucus, that's that phlegm that comes out, right? So after you get better, what happens is you cough and you have this real hard sneeze, and this thicker, kind of chunkier type of phlegm comes out. Well, the reason why we're able to move it is that we're actually able to thin it a little bit with a little bit of saline. And because of the power of our cough and because of the movement of cilia, you're mobilizing it and getting it out of your respiratory system. People with cystic fibrosis cannot mobilize and remove that thick, crusty, kind of full of bacterium and viruses kind of phlegm right there. So what happens with people with cystic fibrosis? The weird thing is the cilia is fine. The weird thing is they don't produce saline, right? People with cystic fibrosis have a problem with sodium receptors on the surface of their cells. Because they can't produce sodium and can't remove it from the cell, you don't produce a little bit of saline. The saline allows you to thin out the mucus a little bit. Thinning out the mucus makes it less heavy, makes it easier for the cilia to move out of your system. When you don't have saline, the phlegm becomes really thick and heavy. The cilia can't move that phlegm at that point. And now it stays in your respiratory tract. It stays in your lungs, right? Eventually, that phlegm starts to, right? I mean, it's a great breeding ground for bacterium, great breeding ground for even fungi, that phlegm. So then it starts to explode in number because you can't get rid of it when you have cystic fibrosis. Us who are healthy, you just get rid of it. There's no time for the fungi, no time for the bacterium to explode in number. But people with cystic fibrosis, what happens is you can't mobilize that phlegm, it stays in your lungs, and now, because you can't move it, the bacterium starts to undergo mitosis, cellular reproduction there, right? And it starts exploding in number and creating really bad infections. And what we see as cystic fibrosis is most people die from pneumonias from cystic fibrosis. The people who do survive actually will also have issues where they usually need to have a lung heart transplant, right? At least a lung transplant to actually get functional respiration to occur. And it's all because of a mutation in the sodium channels, which is crazy, right? So next thing we're going to talk about, besides an intact skin and mucous membrane, remember, we're still talking about innate immunity, are what we call chemical messengers, right? Chemical mediators. I want you to think of these as just chemicals that acts almost like a hormone, right? An enzyme. So some of these we've heard of before. Histamine, interferons, complement, right? These are the ones I want you to know as well. Prostaglandins, a little bit new, all right? Just these in general. Histamine, I want you to know a little bit more because we've talked about it a little bit. Histamine is amine released from mast cells, basophils, and platelets. Now, what is the difference between a mast cell and a basophil? The difference between a mast cell and a basophil, is location, right? So I'm going to come back to this in a second. I believe I have a slide just on histamine. Somewhere here, right here, right? So histamine from mast cells and from basophils. Now, basophils are what we call that cell when it's floating around in circulation in your blood vessels. So when we find that cell in your bloodstream, it's called a basophil. Now, basophils and all white blood cells, those cells really don't do you any good in your bloodstream. When you get a cut, we somehow, if you get a big cut, we know that there's a chance for infection. When you get a cut, there's a chance that you have a microbe there, or microbes, bacterium, fungi, or viruses, even parasites, right, from that cut. So somehow we need to attract your white blood cells from your bloodstream and have it leave your bloodstream so then it can actually go to the tissue that has now been infected. So one of the things that we do is leave little breadcrumbs there. We call it chemotactic factors. What happens is histamine is going to help out that process of movement. All right, we have to move white blood cells from our circulatory system where they're kind of trapped in our bloodstream. And somehow those white blood cells have to find the right location where your infection is and then leave your bloodstream, right? It needs to go to the tissue and actually kill the bacterium that's in the tissue. So it's got to leave your bloodstream. That's one of the functions of histamine. Now, the basophils are the name of the cells when they're found in circulation. Once they leave your blood vessel and goes into the tissue, we call it a mast cell. It kind of changes slightly, but it's basically the same cells, right? There's some small changes, but they're basically the same cells. They release the same granules. They look very similar. The difference is when they're in circulation, we call it a mast cells. When they're in the tissue, when they're in circulation, we call it a basophil. When they're in the tissue, we call it a mast cell, all right? So I kind of reversed it just a second ago, but all right, it is basophil in a blood vessel, mast cell in that infected region, right, in the subcutaneous tissue. So they are the same cells, they release the same material. One of the things they release is histamine. Histamine is a very potent vasodilator. So it causes your blood vessels to dilate. As they dilate, you spread out the cells, forming the blood vessel. So what happens when you spread out the cells? Any gap between those cells also spread out, making it easier for white blood cells to leave your blood vessel and enter the infected tissue. Because remember, your blood vessel will lead to that site of infection, but you need a way for the white blood cells to exit your blood vessel, right? And normally the capillaries, only made out of endothelial cell, with the histamine and the release into the arterioles and the capillaries, you have widespread vasodilation, which causes any gaps between the cells to be enlarged. As the gaps enlarge, right, now whole cells can exit out and go to your target tissue. That's one of the big reasons why histamine is important. Now, if you have, and I'll talk about it later on as well, probably on Thursday, if you have localized release of histamine, meaning that only at the source of injury do you have histamine release, right? That's perfect. That's what we want. We want targeted release in that area. If that happens, we increase vascular permeability, which is a fancy word of spreading out the cells, right? By spreading out the cells that make up your blood vessel, you're increasing the space between the cells, increasing the ability for fluid to leak out and for red blood cells to leak out. We want fluid to leak out because we want more swelling. Remember, the more swelling, the more flow of fluid into the lymphatic system. the more flow into the lymphatic system, the more viruses and bacterium flows into the lymphatic system as well. It gets flushed in to the lymphatic system where all of our lymphocytes are found. Perfect, right? So this vascular permeability allows us to have increased fluid flow out of the blood, right? Capillaries, out of the arterioles, causing edema. It also separates the cells, allowing white blood cells to exit out, which also is important, right? Stimulates glandular secretion, causes smooth muscle contraction in the airways, right? In the bronchioles of the lungs, attracts eosinophils, so then you have more white blood cells traveling to the site of infection. Now, the other ones I want you to know are interferons, complement, and prostaglandins, right? So I'm just gonna highlight the prostaglandins right here as well. Now, with these, right here, right, interferons will be next, right? And then complement. Now, complement, I believe, is the next set of slides right here. Complement is a group of plasma proteins that increases vascular permeability. That's the second one that increases vascular permeability, meaning that we are it is something that we want to achieve, is to spread out the blood vessels, to make sure that fluid leaks out, to make sure that now there's more space for white blood cells to leak out. That's what we mean by vascular permeability, right? Stimulates release of histamine, activate kinins. They also lyse infected cells and lyse bacterium and viruses as well once it attaches. It promotes phagocytosis, making it easier for your macrophage to go to that bacterium and swallow it up. Attracts more white blood cells, neutrophils, monocytes, macrophages, and eosinophils. So then you have more of your white blood cells coming to that site of injury. So in terms of complement, and I want you to think of these as very similar to the clotting factors, right? We had a lot of clotting factors. Those were proteins produced by the liver that were inactivated. A cluster of those proteins. There's like 14, 15 different clotting factors, right? But they're inactive. Same thing with complement. We have these proteins already floating in our bloodstream, even though you don't have an infection. 20 different proteins circulating in our bloodstream in its inactive form. It becomes activated in a cascade formation, and there's two ways of forming it, right? Now, I don't want you to know like all the little niches that we see here. All right, what I do want you to know is the difference between these two pathways. So how do we stimulate the formation of these complement for the activation of these complement proteins? And then more importantly, what does it do? All right, so let's talk about at the very end, what is accomplished when you do activate complement. There's 20 of these proteins floating around inactive, and now we're gonna activate complement. One of the main purposes in activating complement is to somehow fuse all these different proteins together to form something called the membrane attack complex. Now, I know you're gonna, all right, some of you guys will be taking higher level immunology courses. Some of you will be taking microbiology, and they talk a little bit more about it in your class. But in our class, we just don't have time to go to the same detail. What I want you to know then for our class is what is the end result, or what are the end result of complement activation. One of the end result is forming this membrane attack complex, or MAC, right? This MAC right here, membrane attack complex, punctures a hole through the plasma membrane of a bacterium, of an infected cell, right? Now, or the cell wall. By puncturing through the cell wall, the plasma membrane, what we're doing is putting tiny holes in the plasma membrane. What happens then? Well, imagine our cells, the bacterium itself as well, is like a water balloon. What happens, all right, so you have a water balloon, very good analogy to our cells, right? You have fluid on the inside of the water balloon. You're going to have, right, all these different things in our cells on the inside of the water balloon, all right? And what happens if you put a hole in the water balloon? If you put even a small hole in the water balloon, all that fluid leaks out. As it leaks out, the water balloon is no longer be useful, right? Same thing with our cell and the bacterium. As we put little holes, little pores into the cell membrane, into the cell wall, all the fluid on the inside is going to leak outwards. As it does, the cell is going to die. As it dies, you no longer, that cell, that bacterium, that virus is no longer a threat, right? So one of the things it does is to puncture the microbe, so then, and the infected cell, so then you can get rid of it by forming this membrane attack complex. They make channels through plasma membrane formed by classical pathway activation if specific immunity is activated. So we have membrane attack complex, right, activated by classical pathway. Now, let's come back and talk about the pathways then. The classical pathways means that you have an infection, it's already kind of seeded itself in your body, in your immune system, and now we're going to activate the specific immunity, right? So you have a bacterium that's in your body. You've been previously exposed to it by a vaccination or by previous type of infections. What happens then? What happens is that you activate the classical pathway of complement, right? Now, it requires a first instance though, right? It requires a previous infection because once you've been exposed to that virus, you have stored up memory cells. Those memory cells produces antibodies. Those antibodies then is going to bind with that virus or bacterium. As it binds to it, it activates a classical pathway. So there needs to be a little bit more specificity to activate the classical pathway, right? You need to have immune memory. You need antibodies that recognizes your invading pathogen. Only then can you activate the classical pathway and then puncture little holes into the invading pathogen. Now, the other pathway, you don't need that previous response, right? You don't need that vaccination. You don't need to have a previous infection, right? An alternative pathway is part of our innate non-specific immunity. You have an infection for the first time, what happens? That foreign substance or antigen, we'll talk about the foreign protein, the virus is, right? And I will rewrite it here, right? This is the antigen. That antigen, right, comes into your body, comes into your circulatory system. And what we are going to see is that in the alternative pathway, one of the proteins that in complement, a certain one called C3, starts attaching itself to that foreign antigen. As it attaches itself to the foreign antigen, it doesn't kill anything. But what it does do is it looks like it puts a flag there, right? It goes there, puts a flag there, so then macrophages are being attracted to that area. So then it knows that, you know what, this is, because remember, right, you have billions and billions and trillions of cells in your body. So if you have a few 100,000 microbes out of the trillions and trillions of cells, how is a macrophage supposed to figure out that the microbe isn't supposed to be there? Well, one of the ways we kind of make sure that the macrophage finds out that it's not that microbe is a foreign antigen and something harmful is that it puts a C3 protein on it. The C3 protein is like a flag you put onto the bacterium and it marks it and it attracts these macrophages towards that microbe, this bacterium. So then the macrophage can go around, right? It's going to bind to the C3, notices that, well, it's only foreign epitopes and foreign antigens that have C3 attached to it. C3 will not attach to a normal healthy cell. It only attaches to foreign cells or foreign microbes, right? So C3 attaches, macrophage understands that's not supposed to be there, and gets attracted to that region, gets attracted to the cell, and starts to kill it. Perfect, right? So in the alternative pathway, its main purpose is to attract more white blood cells to that area of injury, to the area of infection. By doing that, C3 binding attracts macrophages and stimulates phagocytosis. By putting C3 on there, right, it also attracts more immune cells to that region. So again, if you get a cut, how do we find, right, how does our white blood cell floating around our circulatory system? There's going to be thousands of miles of circulatory system. Many of them are microscopic. How does that white blood cell traversing through the many miles of our circulation, how does it know to get to your arm, right? To that specific area of your arm where there's a cut or an infection. It knows where to go by breadcrumbs, and that's what C3 is. C3 binding to the foreign antigen starts to stimulate and attract more white blood cells to that area. So then it's like a beacon, homing beacon, and now the white blood cells, right, are attracted to that region because that region contains lots of C3 attached microbes, right? So it attracts the cells to the site of infection, also promotes inflammation, and by promoting inflammation, you're producing more edema. So then not only do we have white blood cells attracted to the site of injury, we also have more fluid occurring at the site of injury. So then you'll have more flow of lymph into the lymphatic system. So then the fluid, as well as a microbe, gets flushed into the lymphatic system and brought into the lymph nodes, where you have a lot more T and B cells, a lot more white blood cells there to fight off that infection. So it's a combination of, pretty cool things that's occurring right here. Again, I think it's cool, but it's pretty complex too. So just a little warning here, right? So here, right, this is a classical pathway, right, that starts this process. The alternative pathway can activate C3, then attract more, right, white blood cells through. But the classical pathway helps form the membrane attack complex, which punctures the invading pathogen. Now, the other thing I want to talk about are more almost like immune hormones, right? Immune chemicals. This is a big one called interferon, right? Interferons prevent viral replication, right? So when we talk about a virus is very different than a bacterium. Even though we cluster them together, right? Viruses, microbes, right? Viruses, bacterium, fungi, parasites, even amoebas. We cluster them together as microbes. In reality, they're very different, right? Bacterium can undergo division by themselves, right? As long as there's enough nutrients, they don't need any help, they will grow and multiply. That's why if you have an agar plate and you put some kind of bacterium in there, they divide by itself, right? They don't need any help from our cells. Same thing with fungi, same thing with, some parasites. Viruses, though, cannot self-replicate. A virus is basically nothing more than an external shell that contains all the proteins we recognize. So in coronavirus, you always see a picture of coronavirus and they have little things sticking out of it, right? It looks like little spikes. You have this kind of circular looking virus and little spikes that looks like it's in a corona orientation. That's why we get the name coronavirus. Well, those spikes are viral proteins sticking out, right? Those viral proteins sticking out are actually what our immune cells will recognize. Our T and B cells, our immune cells, doesn't recognize the whole virus. It doesn't recognize what's inside the cell, the virus. It doesn't recognize any of the virus, DNA or RNA. I believe it is a RNA virus. What it does do, our T and B cells are recognizing the proteins on the outside of the virus. Those radiating proteins in a coronal arrangement, that's what our T and B cells recognizes. That's what their receptors will bind to, right? Now, for coronavirus to enter the cell, coronaviruses, any virus, cannot self-replicate. So if you have a virus by itself, it's not going to explode in number. If you put the virus in an agar plate, nothing's going to happen, right? For a virus to replicate, it needs help. It needs our cells. It needs to infect our cells. So right away, we know it's different from a bacterium. Bacterium, as long as there are nutrients and space, it will grow and divide and kill anything that's around it. So then it can produce more of itself. That's how it causes the damage in us. For virus, though, it's different. Viruses have to somehow enter our cells and then take over either our DNA replication machinery or take over our protein synthesis machinery. For the coronavirus, it's an RNA virus. So what happens? It takes over our cells, gets into our cells, takes over our transcription machinery. So then the RNA it has embeds itself in our rough endoplasmic reticulum, and it starts producing viral proteins, right? So by infecting our cell, it starts to replicate itself by taking over our machinery. And that's what makes it so insidious, right? So it gets infected in our cell. Outside of our cell, it's not very, very dangerous. But once it gets inside our cell, it's going to start taking over all the machinery we have to produce proteins, to make more RNA and DNA. So as it does this, it takes away all of our machinery that allows a normal cell to grow and then to replicate. Takes it all over. As it does that, right, our RNA, all right, polymerase, as well as all of our ribosomes, it's taken over by the virus, and the viruses has their own genetic code, and our host, our normal type of ribosomes, then attaches to the RNA, and they start making viral proteins out of it. So the virus's proteins are then going to be produced because they activate our own ribosome and they have their own RNA polymerase. So as they replicate, they take over our machinery. So the most important thing we can do, right, is to make sure the virus doesn't enter the cell. Because if the virus enters a cell, it takes over all the machinery, and that's how they reproduce. So one of the big things that we do give people are interferons. Now, this viral replication is true for all viruses. Some of the retroviruses like HIV, it's an RNA virus. And what happens then is it's got certain kind of enzymes that are found there. And it allows them to turn their RNA genetic code into DNA. Then they have little proteins that actually splices, right, cuts part of our DNA, and it inserts their DNA into ours. So then we start replicating our DNA. In actuality, we start replicating the viral DNA, right? Crazy. So that's the difference in virus. Virus does insidious things like take over our machinery. So then our own cells start producing more and more of the virus. So a couple things then need to happen so then we can fight off that viral infection. One of the first things that needs to happen is we need to make sure the virus has a harder time getting into the cell. Now, coronavirus, the new coronavirus, gets into the cell by attaching itself to a receptor that some of you might have heard of, right? It is a receptor called an ACE receptor, angiotensin converting enzyme receptor. Some of you guys might have family members that are on ACE inhibitors for high blood pressure, right? So that ACE, angiotensin converting enzyme, is found in the lungs. And that ACE then is going to naturally, right, create a kind of immune substance, a substance that causes, right, our blood pressure to go up a little bit. That's the main function of ACE, right, to increase blood pressure. That's great when you have low blood pressure. It's not so great if you already have high blood pressure. That's why we give people with high blood pressure ACE inhibitors, so that we don't produce as much of the ACE, right? Now, with coronavirus, it attaches itself to a receptor meant for ACE. As it attaches up to receptor, then it works its way in through that receptor. Well, one of the things that normally in our immune system, we have interferons. Interferons are like our immune chemicals. That's main purpose is to prevent that virus from attaching and then getting into a new cell, right? It prevents viral replication. Viruses, right, again, needs to be inside of our cell and take over our molecular mechanisms to reproduce itself. So if it's outside of our cell, it really doesn't do any harm. It's when it's in our cell, right? Now, the harm it does when it's outside our cell is that it can infect other cells, right? When it's outside, not too bad, as long as we can make sure that it doesn't get in. That's where interferons come. Interferons are produced by an infected cell, and it causes neighboring cells to produce antiviral proteins. So then the neighboring cells will hopefully produce proteins that inhibit viruses from getting in. Now, some of you might have heard of hepatitis. Most of you have, right? In hepatitis B and C, the frontline treatment to cure hepatitis is to give somebody interferons and antivirals. So have you ever heard of Copagas? Now, next time, right, and I know that they have advertising all the time. They have commercials about it, about hepatitis, right? With the hepatitis medication, right? Next time, look at the real small print. It is a type of interferon, right? So the combination of that medication, the interferon, as well as taking an antiviral, has like a 87% to 88% cure rate in some people. That's a pretty good cure rate for hepatitis. Now, the thing is, you need to take it within a certain time frame. The earlier you take that interferon, usually, I believe the name of the original medication was Copagas, right? The earlier you take Copagas and an antiviral, the better the cure rate. I believe some people that, once they find out they have it and they take it, the cure rate is almost 90%. So it's a pretty high cure rate. Why? Because the antivirals will kill all the viruses that are floating around. The interferon prevents whatever remaining viruses from getting into the cell and causing more viral production. Perfect. Right? Now, besides, so these interferons, once produced and released from a virally infected cell, it diffuses to uninfected neighboring cells, binding to surface receptors. Once you bind to those surface receptors, the virus hopefully can't work its way in. So you can imagine what would happen right now if we can stop coronavirus from binding to this ACE receptor. If we can stop that, the virus can't bind in and work its way into the cell, right? That'll be the best condition. So we will, I think the interferons are expensive, so I don't imagine us using this too often. But what we have done with, with treatment of coronavirus is to use different kind of things that stop its transmission. Antivirals, right? Things like that. Now, interferons are very, very helpful in fighting out that hepatitis. Interferons enhances the activity of phagocytes and a second group of cells that we haven't talked about yet, called natural killer cells. Sorry about that. NK cells. These NK cells, they're not specific, right? So it doesn't recognize a virus as being coronavirus versus chickenpox versus smallpox, right? It just recognizes that there's something wrong and it goes and kills That's what makes the natural killer cells non-specific. Now we're going to see these NK cells, their main purpose is to kill virally infected cell. Very important, right? Keep in mind, all white blood cells need to be able to have what we call, right, immune memory and tolerance, right? One of the things it can't do is kill your normal cell, right? If you have a white blood cell that kills a normal cell, that's an autoimmune disease. That's not good. So one of the things that we need to do is once a cell gets infected, we need to make it okay for normal white blood cells to kill that infected cell. When it's healthy, white blood cell, right, will not kill that infected cell. But once that cell gets infected with a virus, there's no way for that cell to recover anymore. It's going to be producing viral products from that point on. So now, unless we kill that cell, you're going to produce more and more and more viruses. So what we need to do is give it the okay to kill that cell, right? On Thursday, we'll talk about how it does this. But when a virus infects a cell, it will cause changes in the cell that makes it okay, that gives the green light for our natural killer cells to go to that area and kill that virally infected cell. That way, we can hopefully stop the viral replication and viral production. Right? Now, in terms of innate immunity, we had those immune chemicals, right? Histamine, talk about the complement proteins. We talked a second ago about interferons, right? With innate immunity, we talked about an intact barrier, the skin, the cilia as well as a mucous membrane. The last thing is cells, right? Certain cells are part of our innate immunity. Neutrophils, macrophages, eosinophils, basophils, NK cells, right? Those are all part of our innate immunity. Let me see if I have right here, the rest of this right here, right? So these cells, neutrophils, macrophages, basophils and mast cells, eosinophils, and natural killer cells, those are all cells that are non-specific, right? So when a neutrophil goes to the site of injury, right, it goes to the infected site. And usually because of their numbers, because of how many they produce each day, they're going to be the first cells into an infected site. When the neutrophil goes into an infected bacterium, infected area, it doesn't matter if the bacterium is E. coli. It doesn't matter if the bacterium is Pseudomonas. It doesn't matter if the bacterium is Streptococcus or Staphylococcus. It doesn't matter. As long as it's a bacterium, the neutrophil will attack that bacterium. There's no specificity, right? It's not like it recognizes that, oh wait, that's right, E. coli, I can't attack it, I don't recognize it. No, it attacks it no matter what, right? So neutrophils are non-specific because of that. It's non-specific for that specific type of bacterium, meaning that it understands it's a bacterium and kills it. It doesn't matter what kind of bacterium. And that's what makes it general, right? Phagocytic, first cells to enter the site, first cells to die as well. And we see a lot of neutrophils in pus. Now, some of you guys have worked in a nursing home. Some of you guys have actually worked in a hospital for. Eventually, if you're going to be a nursing student, you're going to work with it all. And you will have your own pus stories, right? I will share my pus story next time. But you will go into a room, and right when you go into a room, you smell it right away, right? It can be caused by an abscess, or it can be caused by an ulcer, right? Usually the ulcers are the one that smells, or a leaking abscess smells terrible. And what we see in that abscess and that ulcer is just that fluid, that white, thick fluid that is pus. And that is a combination of neutrophils and dead bacterium, right? It's pretty gross. Take a look at the number. 126 billion neutrophils are produced a day. Billion, right? So if you think about our our red blood cells, right? We produce 2 million every second, right? We're going to produce literally hundreds of billions and trillions of red blood cells a day, right? Again, right, about 1.7 trillion red blood cells a day would be produced, which is crazy, right? And 1.26 billion neutrophils only, and they're active against bacteria. Macrophages are seeing about 20%, right, of our, CBC with diff. Large phagocytic cell, when they're floating around your bloodstream, right, they are called macrophages. Monocytes then leave the blood and enter the tissue. Now, we have certain, kind of like how mast cells are called mast cells, but when they're in subcutaneous tissue or in a specific tissue, we call them mast cell. When they're bloodstream, we call it basophil. Same thing here, right? Now, when we find macrophages in our skin, it has a specific name, Langerhans cells. When you find macrophages in your brain, in your central nervous system, we call it microglial cells. When you find it in the liver, we call it dendritic cells. So all of those are types of macrophages though. And they do the same thing. It's just that they have a slightly different name to it based on location. The main purpose of the macrophage is to destroy, right? Eat up any of the bacterium, virus, or fungi that's around, that's causing an infection. Secondly, once it kills it, it's going to display it, right? So it kills off a bacterium. As it swallows up and kills it, all of the different proteins on the outside of the bacterium, on the surface of the bacterium. So those proteins on the surface will then attach to receptors on the macrophage. As receptors attach to all the different proteins, thousands of different types of proteins found on the surface of the bacterium, you're going to have a combination of protein from the bacterium and receptor from the macrophage. And now you're going to put it into the cell surface of the macrophage. So then you can display it to other cells. So then you can not just destroy the bacterium, but now you display it to other cells. So then you can activate other memory cells. So then not only do you kill that one bacterium, you can activate other cells that can kill even more bacteria. Perfect. Basophils releases histamine and heparin. Mast cells are the basophils when they're found in the connective tissue. Eosinophils secrete enzymes that kill parasites. All right, and there's a big one right here. There's the only one that's different. We've talked about the other ones. NK cells, natural killer cells. Even though they're a type of lymphocyte, they're non-specific. They can lyse tumor cells as well as virally infected cells. Now, when we mean by non-specific, it means that it doesn't matter if this infected cell is infected with varicella zoster virus. Doesn't matter if this infected cell in our body is infected with measles or mumps or coronavirus. As long as you have a cell that's been infected with any virus, it causes the cell to slightly change. As it causes the cell to change, NK cells can go around and kill those cells now, right? And we'll talk about how, again, on Thursday. Now, these are also the cells that are incredibly important in how we fight off some kind of tumors and cancers. We're going to see that in the future, right? When you give somebody a chemotherapy, radiation therapy, they kill all actively dividing cells. So we know that in a tumor cell, in a cancer cell, right, they're dividing all the time, right? Because they're dividing all the time, they grow much faster than a normal cell. What happens? Because they're growing so much, if you give somebody that has cancer chemotherapy, right, they're growing so much, they're going so many rounds of replication that the tumor cells pick up a lot more of the toxin, a lot more of the chemotherapy. So actively dividing cells pick up the chemotherapy. So the tumor cells that are actively dividing pick up the chemotherapy and they die. The problem is it's non-selective, right? In your GI tract, you're going to have cells that are actively dividing, right? Your hair follicles have cells that are actively dividing. So those cells also pick up a lot of the chemotherapy. And what happens when you're under chemotherapy? Hair loss, because you kill out those follicular cells. And in the GI tract, nausea, vomiting, as well as diarrhea from picking up excess amount of chemotherapy. In other words, it's not targeted. You're killing all actively dividing cells, even the healthy actively dividing cells. By activating the NK cells, hopefully in the future we get better at it, we can hopefully target cells that are changed. So then these NK cells can target, right, only cells that have mutated, right? And they will only kill those tumorous and cancerous cells. Right. Now, we'll stop after going through this. I know there's a lot. We'll go through information. I will save adaptive immunity for Thursday, just because it gets really, really intense. And I know after a couple hours of listening to this, you guys are getting kind of tired. So we'll save that for Thursday. Now, in terms of this inflammatory response, keep in mind, right? And again, with natural killer cells, we'll come back to it in a second, right? Now, with inflammatory response, tissue injury, regardless of type, can cause inflammation. In other words, whether it's caused by a cut, right? Whether caused by infection, it can cause inflammation. Response to initiated by chemical mediators, including the big one being histamine. Histamine produces vasodilation, produces increased vascular permeability, right? And we have complement hormones and proteins that have chemotactic attraction, right? Now, fibrinogen complement enter the tissue. Fibrinogen converts to fibrin, walls off the infected area. Complement enters the site and starts attaching itself to the microbes, right, C3, and it starts to bring more white blood cells. Now, in terms of inflammatory response, there's two different types. One that's local. You get a bug bite. If you get a bug bite on your arm, only that area where you get a bug bite should swell up. That's a good response. It's localized. Only the area of damage do you have that enlargement, right? Symptoms include redness, heat, swelling, pain, and loss of function. Now, sometimes you go beyond a local response and you get a systemic response, right? And sometimes a systemic response isn't bad. Other times it can cause us to die, right? So the systemic response of inflammation is when many parts of the body gets activated. Same symptoms as local, but more widespread. Cause increase in neutrophil count and numbers released by the red bone marrow. So this is a positive, right? You also produce fever due to the production of pyrogens by various kinds of cells. All right, what we're gonna see is later on, when we talk about interleukins, they produces, right, they're immune hormones that activate other type of T and B cells. But those interleukins are a type of pyrogen. It increases your body temperature to 101, 102 degrees. Now, the weird thing is this. All of our immune chemicals, complement, histamine, interferon, interleukin, all of our immune hormones work better. And even our white blood cells work better at a higher body temperature, 101, 102 degrees, right? The problem is we've all had a small fever before, right? A very low grade fever. Even if your body temperature is like 100.5 degrees, you feel pretty bad, right? You feel sluggish, you feel tired, you feel lethargic, right? You might even get the night sweats from it. In other words, even if it increase the body temperature, right? Normally 98.6. If you increase it 1.5 degrees to 101, even that makes you feel bad. Now, the good news is that it actually is more effective in terms of our immune system. Higher body temperature makes the cells more active, the immune cells, and increases the efficacy of our immune hormones like interferon, makes it better, right? The problem is we don't feel good. So we don't want it to really reach that level, 100, 102 degrees, unless we're really sick. Now, this is also the reason why, and we're going to see in a second, and next on Thursday I'll discuss it more, why doctors always tell you, especially if you have a child, right? Or tell your mom and dad when you were kids, usually do not call the office unless the fever is at 101 degrees and above. Why? Because usually when it's below 101, it's caused by our own immune system fighting off a viral infection. Now, there are some viruses, like coronavirus, like swine flu, that causes it to go beyond 101 degrees. But during a normal kind of, the common cold infection and some of the flu infections, it doesn't go beyond 101 degrees, right? Normally, if it's above 101, it's 102, 103, it's caused by bacterium, right? So when you have a child with 102-degree fever, we call the doctor almost immediately prescribes antibiotics. Why? It's because usually that's caused by that high-grade fever caused by a bacterium, right? I say that right now, but now with coronavirus, we know people have been known to have long-term spikes in their fevers for days, weeks on end, right? And even a few weeks where the fever is at 102, 103, right? So there are some very virulent viruses that does cause you to spike a fever even higher than normal. Now, what does the inflammation do? Right? Widespread increased vascular permeability due to systemic release of histamine can cause, right, large volume of plasma to enter the interstitial fluid, leading to shock. It's almost like an anaphylactic shock reaction. So when you have localized immune response, localized inflammatory response, it's almost always good. When you have systemic inflammatory response, some parts of it are good. Fever, increased neutrophil production, but some part of it can cause, right, anaphylactic, right, shock to occur. Natural killer cells kill a variety of microbes, tumor cells. They're very important for immune surveillance. By the way, these NK cells, these are the main cells that will then, if you've ever had or your parents need to do an organ transplant, right? Kidneys are the ones that we think about most, right? If they need kidney transplant, you've heard of people getting a rejection of the kidney. It's because of these cells, right? When you have a kidney rejection or any organ rejection, it's because the person's immune system, the person that's receiving the new kidney. Well, unless you have an identical twin laying around, most of us don't. If you have an identical twin, every single receptor, every single protein, every single cell should be identical because they have the exact same DNA, right? When we compare one twin to the next. But besides that, right, if you have a fraternal twin, let's say, a fraternal twin is produced, right, from the release of one egg and one sperm cell, right? So you have one egg, one sperm cell should produce a child, right? You have fertilized egg. And then you have a second egg and a second sperm cell, right? For some reason, you ovulate out 2 eggs that month. So now you have a separate egg and a separate sperm cell also being fertilized, right? So in a fraternal twin is 2 separate eggs, 2 separate and different sperm cells. So they're going to be just like any brother and sister. What makes them twins is that they're born on the same day. That's it. But genetically, they're just like any other sibling, right? Identical twins are not formed that way. Identical twins are formed from one egg that's ovulated, one sperm cell, right? And now you should have just one baby. But for some reason or another, during the process of cell division, instead of forming different parts of the same baby, you form two different babies. What happens then? Well, the two babies come from that original, fertilized egg. They have the same genetic material, right? All of the DNA should be exactly the same. So, can you have an identical twin that is of different sex? Can you have one male son, one female daughter, and they're identical? No, right? There's no possible scenario where that can happen because they came from the same sperm and the same egg, right? These are true twins. The DNA is completely shared and identical to each other. So if you have an identical twin, you can take the twin's kidney and replace it with the other twin's kidney in the other twin's body, and there should never be any rejection. But most of us don't have this, right? Instead, we try to find a close match. Somebody, we're going to match their, right, what we call their HLA, right? Match the different type of receptors, make sure there's somewhat of a match, but there'll never be a perfect match. Why? Because what we're going to see is that we have what's called MHC1. MHC1 are receptors that are sticking out of all of our cells that have a nucleus. All of our nucleated cells have this receptor, MHC1 receptor. It's basically a receptor, and on that receptor has a little bit of our own DNA, our own protein that's on that receptor. It's like a flag that says that this cell is part of my larger body. Now, what happens when I give somebody a different, right, organ, a different kidney? Well, what happens with a different kidney is the kidney and the cells of kidney have different flags, different MHC1s, right? Because it's not for me, and I don't have an identical twin. So the actual cells that make up the kidney have different receptors and, right, protein sticking out. Protein receptors combination that's different than mine. These NK cells will then go to that site, realize the new cell is slightly different, and starts to kill the new cell. As it does so, it gives us the kind of rejections that we call host graft rejections, right? So these NK cells are very important in that it gives us that ability to kill tumor-infected cells and virally infected cell. What we're going to see is when a virus infects a normal cell, it changes the cell. It changes the receptor, MHC1 receptor, on that cell. All right. Normally, for me, right, on all my cells that have a nucleus, all the cells in my body with nucleus have this receptor, MHC1, and on the receptor are proteins specific to me with my DNA tag on it. That means that this cell is part of my larger body. When that is accurate, the NK cells leave that area. Doesn't kill off that cell. But when you have a virally infected cell, it changes our receptor protein combination. Because remember, when you have a virally infected cell, you're producing viral proteins now. So the viral protein attaches on this MHC1 receptor. What was my protein now has virus proteins on it. So the flag is changed. As a viral protein is then expressed, NK cells note that change, and now it has an okay to kill that cell, which is perfect. Now, tumor cells, they start to grow unchecked. As it starts to grow, their MHC1 also changes. Hopefully, then it can kill out that tumor cell. All right. Any questions so far? No? All right. So I will end it right here just because I want to pick up on Thursday talking about fevers and inflammation a little bit, and then go and talk about specific immunity. All right. Specific immunity is very intense. So please read that, and we'll try to finish up within that first hour on Thursday. So then we can talk a little bit about the respiratory system on Thursday. All right. So 3 people dropped off on that lecture. I know it's a tough lecture. All right. Any questions?